Our next speaker is Jason Stetler. Jason is a research scientist with the Utah Division of Wildlife Resources in cooperation with the Great Basin Native Plant Selection and Increase Project. His primary research areas are native plant material development of forbs native to the Great Basin and developing agricultural practices to improve germination, establishment, and seed production of these native forbs. Please welcome Jason.
All right, so some of this research was done by me and some of it was done by Kobe Jones, who was my predecessor in this project. So if there's any problems, it's probably his fault. So what we work a lot with native forbs for plant materials development, and then also a lot of the problems with that and getting enough seed for restoration purposes is having growers grow it. And the main problem with that is there's a lot of risk involved with a new species or a new crop because it could fail, lots of things could happen. So we try to do research to kind of minimize that risk for the growers. That way they'll be producing more seed. All right, so we're working with lupines here. They're an important component of ecosystems, and so we have 4 lupines that we work with primarily. First is Lupinus argenteus. It's an indeterminate flower seed set, so at any given time when you harvest the seed, you'll get about 30% of seed. And if you're a grower, that's a problem because you're going to want to mechanically harvest so you can maximize your seed set. Otherwise, you're going to be hand harvesting, and that drives the cost way up. It has the smallest seed size out of all the species we're doing research with. It's about 80 seeds per gram, but it's the most widely distributed. The second species is Lupinus arbustus. It has determinate seed flowering and seed ripening, so it's pretty good for farming and that kind of stuff because it's going to, well, most of the seed will be ripe at the same time. And it's a yellow flowered species, which is pretty, and most people like that. But it is one of the least abundant ones, and it's mainly right here in the Great Basin, a little bit up north, probably towards the Columbian Basin area. The third species is Lupinus prunophyllus. It also has determinate seed set and flowering, so it can potentially produce a lot of seed and you'll have to make one harvest of it. The problem with the distribution of this one is some people classify it as a subspecies or variety of Polyphyllus or Washington lupin, and so when we go through the herbarium data sets, it's hard to tell which one is what. So it has a more broad distribution, but I could only use ones that said Prunophyllus in the species, just to be sure I wasn't including stuff that was not in there. And the fourth species is Lupinus sericeus. It is indeterminate, as you can see here. Some of it's already going to seed, and others just starting to flower. So it's very variable in when it produces flowers and seed. So the first part of Kobe Jones's project was to study seed dormancy in these 4 species. And it's primarily a physical seed dormancy because it has these species have thick seed coats. So he, there's a map of distribution. So he had several different treatments for them. The first 2 were thermal scarification, so constant temperature, and he varied the time. So he had water at 100 degrees Celsius, and varied time was 15 seconds through 60 seconds in 5-second intervals. Second, thermal scarification was varying the temperature. So we started out at 50 degrees Celsius, and it went up in 5-degree increments to 95 degrees, and time for that was 60 seconds for each one of those. Then the chemical scarification with sulfuric acid, and that was between 1 and 10 minutes in 1-minute intervals, and then sodium hyperchloride, which was 15 minutes and 15-minute intervals all the way up to 150 minutes. And then the last one was mechanical scarification in a phosphorus batch scarifier. So it's basically sandpaper on the outside drum and these blades inside, push it around and scarify it that way. That saves a lot of time because a lot of scarification is done by hand, so that's pretty good for that. So then he put them into Petri dishes with some blotter paper and put them in the ink and let them germinate just on the countertop in ambient conditions. So with our argenteus, the 6-minute sulfuric acid treatment produced 82% germination rate, and the 4 to 6 second mechanical treatment produced 84% germination, and thermal scarification and sodium hypochlorite didn't make any significant difference in germination rates. Arbustus, no treatments increased germination, but all significantly decreased germination over the control. Chlorophyll, the 5-minute acid scarification produced 80% germination, and the rest were insignificant. With sericeus, the 3-minute sulfuric acid produced 85% germination rates, and 4-second mechanical produced 75% germination rates. So there's a lot of variation in there, but through his study, what he did, he showed that thermal scarification made no difference, and using the hyperchlorite versus chemical scarification didn't do anything either. So the next step in his research was for planting, for cultural practices. So there were 2 methods he used, broadcast drilling, and then he covered it with some sawdust and then insulate fabric, and then just direct drilled seed in and didn't cover it, didn't put a mulch treatment. And the reason why he did the mulch treatments was to improve germination or stratification period. The mulch holds in the moisture and protects from frost damage and stuff like that in the spring. So as you can see from the field, some did really well and others not so much, and some nothing happened at all. So for germination, the basic trend for all of them is the ones that were covered with the mulch had an increased germination rate. And then for establishment, obviously the ones that had the better germination established better. But Lupinus arbustus and Chlorophyllus had iron deficiency chlorosis really bad, and arbustus just completely died off right after it germinated. And then we wanted to look at seed production. There's 2 methods, mechanical harvest and then hand harvesting. Mechanical is obviously easier, faster, you get it done better. But with some of the species, if it's indeterminate seed ripening, you're going to want to do the hand harvesting to maximize your seed yields. So we have the 3-year data trends here. With argenteus, the first year it performed the best. And sericeus, didn't do much. And 18 pounds an acre isn't much if you're a grower and you want to grow this. You're not going to make much money off of that. But after, by the second year, the sericeus did the best, and the 3rd year it was also the best. And as you can see, argenteus peaked at 2 years and then decreased after that. But with these, with the harvest data, all the plots were harvested and averaged together. So with argenteus, there were no plots that were from the uncovered treatment. All those plants died off by the seed harvest. But with sericeus, there was both of those. And if we used both, let's see. If we calculate the covered versus uncovered separately from 2011, the sericeus uncovered plots average 145 pounds an acre, while the uncovered average 42 pounds an acre. So there's quite the variation there. So if you're a grower, you want to grow some lupin, obviously the cover's going to have a lot better chance for you. And with the Prenophilus, it had the iron deficiency chlorosis, and it really stunted its growth and development. And by the 4th year after planting, it finally flowered and went to seed. And there was a lot of variation within all the different plots. It ranged from 11 pounds an acre to 167 pounds an acre. So that would be a nice one to have if you can figure out how to get it to grow evenly across your whole field. All right, and so conclusions from his studies were Lupinus, arbustus, and Prenophilus are extremely susceptible to iron deficiency chlorosis. The use of insulated fabric improves germination, establishment, and seed yields. And Lupinus argenteus is productive for the first 2 years and then will only be replanted. And Lupinus sericeus is productive for at least 4 years. And so then when I came into the project, we wanted to address the challenge of iron deficiency chlorosis. Iron is one of the most abundant nutrients in the soil, but it's immovable in the plants. And lots of plants experience iron deficiency chlorosis in high pH soils. There have been lots of studies in Australia with lupines. It's a huge cash crop there, over 1 million acres of lupine crops planted in Australia annually. And with their research, they found that the plants exhibit iron deficiency chlorosis in high pH above 6, and that's still acidic soil. So on our farms, our soil is around 8 pH level. So the challenge is, how do we treat that? There have been studies conducted in the Midwest with soybeans, and they've used different applications of soil iron treatments, foliar iron treatments, and seed iron treatment. So we decided to try that here. And there are 2 tolerant species, argenteus and sericeus. They didn't really exhibit many symptoms of iron deficiency chlorosis, but arbustus and Prenophilus did. So we took these 4 species and we added these treatments, a control, so we didn't do anything to them. We applied seed fertilizer to the iron fertilizer to the seed, or we sprayed it in the spring, or a combined seed and foliar application of the iron fertilizer. And then the measurements we take are germination rates, establishment rates, and then chlorophyll content and fluorescence ratio with a chlorophyll content meter, seed yield, and plant height at maturity. So we had the iron chelate here, but you need to get it to stick to the seed, and so we used a product called M Binder. It's the seed coat of a Plantago species. They grinded that up, and it acts really mucilaginous. I can't say it. Real snotty, so it sticks everything to the seed real well. So it holds in the moisture and it holds on to the iron treatment, and then we also added the rhizobium inoculum. We let that air dry, and then we seeded it with a precision seeder, and we covered those plots with insulate fabric because that produced the best results. You can kind of see some are yellow, some are green, and some aren't there. So germination, the Lupinus argenteus didn't grow at all, and really the germination rate isn't going to be affected by the different treatments. We just need that as a baseline for when we come up with the establishment rates this year. So then we took readings of the plants with a chlorophyll content meter. We randomly sampled 20 plants per species and treatment in all of our plots, and then we correlated that with everything else. So we can look at the results. Some, you can see here, some are pretty robust plants. Some aren't, some are little, and that's what we expected because the sericeus was tolerant to iron deficiency. So we look at Lupinus arbustus here. The control, the foliar application, the seed, and seed and foliar. It's kind of hard to tell the difference visually. Just looking at it, you can really tell the difference between all of them. A lot of these iron deficiency chlorosis studies are done with visual ratings, 1 for green and 5 for yellow. But with the with the chlorophyll content meter, it gives a more accurate display of what's actually in the leaf itself. And we also did a native populations of these species to kind of get what it should be in its natural settings. So we have both two. So both, it has the seed treatment, and so we measured that before we put the iron application, and the 2 is post-application of the foliar iron. Same here with foliar, so this is pre-application, so basically it's the same as control. And then the foliar is post-application. And doing the combined treatments of seed and foliar iron, it basically is significant. The difference between that and the native stand is the same. So there's no statistical significance between the two. And then also from that in the foliar spray. And it's basically the same thing with the fluorescence ratio or the chlorophyll. It shows the same trend line. But if we'll pay attention to this one right here, the native stand, the mean values for all 3 species are approximately the same. So that's also good to look at. So here's the Prunophilus. It was medium susceptibility. Control, foliar, seed and foliar. You can tell a little bit better with yellowing and green, but it's still very variable across all of them. So the combined treatment and the foliar treatment were about the same. Oh, and the seed treatment were about all the same as native. There was no significant difference between those 3. But obviously the control and was lower than all those. So we look at sericeus, and you can tell there's not much difference in green, as you can see visually, but there is a difference in plant size that we noticed early on. So we're going to, when we measure the plants at maturity, we'll see if that correlates all the way through. But if you look here, the native chlorophyll concentration is less than the combined iron and seed and foliar treatments. But it's about the same for the foliar treatment. So, and this kind of reflects what the study's been done in soybean research. They found that putting the iron applications on tolerant varieties of soybeans actually increases seed yield better than it does susceptible varieties. So we can see here that it's pretty good treatment, and well, this was the first year of the study, so now we just have to keep going on. So the conclusions: argenteus failed to germinate. Seed application of iron significantly improved chlorophyll content with all species. A combination of seed and foliar application brought chlorophyll content to the level native stands with all species. And sericeus had the greatest increase of chlorophyll to all treatments. So the next step is this spring we're going to take new chlorophyll content reader meetings and see if that correlates through 2 growth years. And then evaluate the seed production and plant height at maturity. We're also going to do some greenhouse studies studying biomass. It's really hard to get the plant biomass and root nodulation from a field setting. It's hard to dig up the whole plant and make sure you have it all. So we're going to do that controlled in pots in the greenhouse and see if we can get a correlation between biomass and nodulation for the iron treatments. And that's all I'd have. I'd like to thank my funders, Great Basin Native Plant Selection Increase Project, and Forest Service, and WLM.
Question for Jerry.